The structure of chromosomes pretty much has a pair of sister chromatids. These chromatids are attached to the centromere. So the entire chromosome, which is pretty much DNA wrapped around proteins called histones, and sections of your chromosomes are called genes, which characterize certain specific features. Now, how does an organism, for example, in mitosis, one cell produce two daughter cells which are genetically identical? Well, we first have to discuss the cell cycle. The cell cycle has interphase. Interphase has the G1 phase, the S phase, and the G2 phase. The cell spends most of its time during interphase. The mitotic phase is where the cell actually divides. And so the G1 phase, cell is growing. The S phase, DNA replication occurs. And then you eventually have your G2 phase where the cell is getting ready for mitosis. Now, mitosis actually has certain stages. So we will talk about the stages of mitosis. Mitosis, first of all, when you can see the chromosomes or the chromatids, then that is prophase, when you can actually see the chromosomes. Metaphase is where the chromosomes are lying on the equator and connected by the microfibrils to the centromere. So notice they line up middle, on the middle equator. Anaphase is where the chromatids start moving towards the poles. So here you see chromatids moving to either end. And then finally, telophase, you get two genetically identical cells. Now, what happens after that? Cytokinesis. That is division of your cytoplasm and complete formation of two cells. Now, in animal cells, you have a cleavage furrow forming, but in between cells, but then in plant cells, you have a cell plate forming between these two cells during cytokinesis.